Hello, welcome to 13 Universal. This is Shedro, okay, and we are looking at A-level CI biology again. This time around, all topics paper 4. This is A2 syllables series 2. We have done series 1. Find it and watch it, okay? Share it with your friends. Get your writing materials as usual. Let's practice some concept. All right, now guys, we're going to be looking at bulky background information. You have to read information and remember what it says. So the testing your ability to apply, be able to learn something new in a short time and then apply learn concept in an unfamiliar context. All right, so let's go. You're going to be seeing what you've not done before. Exactly. Say the potato plant Solanum tuberosum is an important food crop. Crop yield is reduced if the leaves of the plant are eaten by larvae immature stages, okay, of Colorado beetle Leptinotarsa des lineata. Okay, so it's talking about a particular all right plant. So the next paragraph, crop scientists use recombinant DNA technology to create two genetically modified GM varieties of potato plant. These plants produce protein that are poison, proteins that are poisonous to insects. Okay, so they produce proteins that are insecticidal. All right, let's look at them. GM potato variety A contains two genes SN and BT. GM potato variety B contains two new genes SN and OCII. Okay, now next paragraph, these are bulky background information, but it's okay, you know, just read it and try to remember it, understand it. So the new varieties were tested by having a constant number of Colorado beetle larvae introduced to plants at time 0 hours. The number of larvae that were alive after 24, 48, and 72 hours was recorded. Okay, the percentage of the larvae that had died in each time interval was calculated. This was repeated for potato plants that had not been genetically modified, non-GM. Okay, so that is the information. I hope you can still remember this. My advice, my recommendation is to go over it again and read it. Okay, understand what it says before you go into questions. All right, so that's background information. Let's dive into the question. If you have paused the video and go over it, let's dive into the question. It says table 4.1 shows the percentage of Colorado beetle larvae that had died on GM potato plant varieties and on non-GM potato plants. Okay, so we have genetically modified potatoes that can make poisonous proteins against insect larvae. All right, let's see what I have there, guys. Jump with me. That's the table. Type of potato, this is GM potato variety A, GM potato variety B, and non-GM. Right? Why did they put non-GM there? All right, it's for control. It serves to compare baseline, right? Okay, whether this thing works. The first question there, remember, you must be able to understand the background information and you must be able to retrieve the content. We must match the content to suit the context. All right, this is a skill. Now look at it. Suggest what is meant by recombinant DNA technology. So what is meant? Suggest means give us a logical, explanation of what recombinant DNA technology is. There's no one valid. Only it has many valid, all right, responses to it. That's why it's what suggest means. Now, for me, recombinant DNA technology is just a process where DNA molecules are joined, DNA molecules from different sources. All right, it is a process of joining, of combining different molecules from different sources. Okay, using enzymes, like restriction enzymes used to cut them, and then ligase enzyme used to join them, and then they are transferred into another organism that now becomes genetically modified or transgenic. So it's a process whereby two DNA molecules from different sources, all right, are joined together and transferred into another organism, all right, of the same or different species. That organism now becomes genetically modified. That's recombinant DNA. So transferring recombinant DNA. Recombinant DNA is a DNA with, all right, molecule DNA, different DNA molecules from different sources. Okay, so let's look at what the answer E says. Just 2 marks question. It says DNA join. You must mention, these are the points that must be in your answer. You must mention that DNA molecules are joined, all right, or combined, made of 2 different sources using named enzyme. Okay, named enzymes, there could be the restriction enzymes, okay, which are used to cut them, and the ligase enzyme, which are used to join them to form sugar phosphate backbone, and that, joining DNA and the vector, which is also made plasmid, and then non-transmitted transferred to the organism that becomes GMO or transgenic. Is that okay? So that's just 2 marks. All right, let's go to the next question, guys. Yeah, says suggest why the scientists created 2 different types of GM potato plant. Why did they create 2 different? We have GM potato A and GM potato B. Why? Okay, the reason, of course, is to compare these 2 genes. Okay, to compare gene A and gene B, or to compare their proteins, to know which one works better. All right, which one works better, whether it is A that works better or B that works better. All right, okay, so now the answer says to test or compare effectiveness of genes, proteins, BT, all right, okay, and OCI plant variety A and B, to see which gene plant variety of A and B works better or best. Just 1 mark question. Okay, let's look at the next question. Remember, we're solving this together. This is same table. State why the scientists also perform tests on non-GM potato plant. Okay, write the answer down. Pause the video and write the answer. Why does it have to bring non-GM potato? I mean, we could have been okay with this, right? Now, this non-GM potato, right? Why? State why. Okay, and just how many marks question, right? It's just 1 mark question. Okay, so why, of course, is for control to compare with the GM plant. Okay, to serve as control, so to compare or provide a baseline, okay, as a control. 1 mark. All right, let's go to the next question. The same table. Now the question says discuss how the results in table 4.1 provide information that could help solve global demand of food. How does it, okay, provide information for that? Okay, so what does this tell us when they say discuss how the results in table provide information that could help to solve the global demand of food? So what does this tell us, guys? All right, what does it tell us? Does this solve global demand of food? Some people are dying of food in some part of the world. Now, if we make GM potato for them, potato is a staple food, right, that most people eat. It's a basic supplies, carbohydrates, energy, all right, for activities in the body, cell activities and all of that. Now, yes, can you see that both of them, the percentage of Colorado beetle that had died, you see that they kill, both A and B kill the larva. They kill the larva. This is percentage of Colorado beetle larva that had died. So after 24 hours to compare 2 of them. But you see both of them, generally, this is general trend, they kill the larva. All right. But if you check, you see that A, GM potato variety A, kills the larva more, because after 72 hours it had killed 100 of them. Okay, now when they kill the larva, they increase yield. There's increase in potato yield because the larva reduces harvest, reduces yield. So if you kill the larva, the yield will increase, and once the yield increases, we have enough food, all right, to supply to the demand. Is that okay? So let's see what they have to say. Look at it. A and B kill larva. That's what this tells us. It kills it. That's what this number tells us. Yeah. Okay. So GM increases yield. By killing the larva, yield is increased. And don't forget that A is best or kills or works faster because, can you see? 100 over 93 against 93, it kills it more. Or provide most food. Okay, so that's 3 marks there for you. Let's go to the next question, guys. Number 5, the respiratory quotient RQ values for different respiratory substrates can be calculated. All right, table 5.1 shows the formula of 4 respiratory substrates, the number of oxygen molecules oxygen needed to completely respire each substrate to carbon dioxide and water. So look at it, each of them, guys. Respiratory substrate formula and number of oxygen molecules needed for respiration. Then also respiratory substrate with the highest RQ value, respiratory substrate with the lowest RQ value. Now, guys, I want to show you something. Okay, remember, you remember the formula, but pause this video and try the question first. Pause it. Pause the video and try the question. Remember that RQ value, right? RQ is equal to, remember what it is, all right? Number of moles of CO2. Let me use it as number of moles of CO2, N of CO2, okay? Number of moles of CO2 or number of volume of CO2 formed all over number of moles of oxygen, which is O2. Remember this formula, guys? Remember it? This is the formula for RQ, if you want to calculate it. So automatically, this means that RQ, right? RQ is inversely proportional, okay, to number of moles. That this is that is not NO2, it's not nitrogen oxide, it's number of moles of oxygen, okay, number of moles. All right, this is number of moles. N means number. This is also number, right? Yeah, it also means number. Okay, yeah, that's an N, right? N. So that means that as RQ value, as number of oxygen, number of moles of oxygen increases, the RQ value decreases. So they said respiratory substrate with the highest RQ value, the highest RQ value will be the one with the lowest number of oxygen molecules needed based on this relationship. Okay, and that one that should give us the highest RQ value is malic acid. So that suggests also the one that will give us lowest RQ value is the one that has the highest number of oxygen molecules. So the one that has the highest oxygen number of oxygen molecules is this. That's 25.5, and that's oleic acid. So this is malic, lowest. Okay, that's highest. This is, of course, lowest is oleic acid. Okay, oleic, linoleic. All right. Yes, so you see that, guys. So let me clean up this. You can pause the video and watch it again and share this. So the highest malic, lowest oleic acid. Just 2 marks. All right, let's quickly run to the next question. So in an anaerobic condition, the production of ATP in mammals and yeast involves glycolysis and fermentation. Describe the similarities and differences between fermentation in mammals. Now, fermentation in mammals is what? And in yeast. So pause this video, okay, and tell me the difference between fermentation in mammals and, all right, and their similarities, and that of yeast, the differences and similarities between fermentation in mammals and in yeast. In another way, they can ask you to compare them. They can remove similarities and differences and say just compare, right? Compare this and this. So the same way, okay? So first of all, you've got to pause the video and try it, and then we'll look at the similarities, okay? Remember, the similarities there is that they are both fermentation process, but you see, where do they occur? They both occur in the cytoplasm or cytosol, okay? So let me write something. Now look at the first one. This is pyruvate, all right? So when I write it, you tell me which one is that. That's pyruvate. Is broken down, first of all. Remember, pyruvate is 3-carbon compound. So the first thing that happens is it is converted, right, to ethanol. Ethanol is 2 carbon compound. Remember, ethanol. Okay. Ethanol is 2. Now, guys, what type of reaction is this? All right, this is, of course, CO2 is formed here. That's the decarboxylation, removal of carboxylic group in form of CO2. Ethanol is then further reduced, okay, to ethanol, right? So ethanol, so this is oxidation, reduction. Ethanol is reduced to form ethanol, okay, the same 2 carbon. Now, of course, you can see that. Now, how is it reduced? NADH donates hydrogen. Remember that the role of this is to just dump hydrogen to regenerate NAD so that glycolysis can occur, can continue. All right, that is the reason for fermentation. So this is gonna, okay, so we're going to regenerate our NAD. You know how this works, guys. NAD, and of course that the hydrogen is added, okay, to ethanol to form ethanol. All right, this is one of them. There's another one here, which is pyruvate. Remember, pyruvate also undergoes that fermentation. So what do we have? Oh, that's pyruvate here. Okay, hope you're enjoying this class, guys. All right, now this, all right, is just one step, but it is reversible, guys. That's interesting. So we're trying to recall concept on this, and you just answer question. Okay, so it's going to give us lactate. So pyruvate is converted to lactic acid. Okay, so that's just one step process here. And it is reversible. Okay, now both of them have 3 carbon compound. Of course, here we're also adding NADH. So we're adding hydrogen, right, donated by NADH, so that NAD can be regenerated. You remember? So we need to generate NAD so that glycolysis can continue. So NAD is gotten back. Remember, we have it back, right? Hydrogen is donated to pyruvate, so pyruvate is reduced, okay, to form lactate. This hydrogen I'm writing now is donated to pyruvate, so that NAD is okay. Now, guys, so which one is the process of fermentation occurring in mammals? Okay, so of course, which one is this or this? That is this, pyruvate. So in mammal, can you see now the similarities is that similarities are: number one, both of them occur in cytoplasm. Okay, number two, both of them use pyruvate is used in both of them. Okay, number three, both of them regenerate NAD. Can you see? NAD is regenerated in both of them. All right, and both of them involve redox reaction. Okay, so NAD is undergoing of course NAD undergoes oxidation, loss of hydrogen, while ethanol is undergoing reduction. The same thing here, pyruvate is undergoing reduction, NAD is undergoing oxidation. Okay, so I've mentioned 4 things. They both involve pyruvate, they both occur in cytoplasm, they both regenerate NAD, and they both involve redox reaction. Okay, but what about differences, guys? See, this produces 2 products, CO2 and ethanol. This produces only lactate. Okay, all right, this is just lactate. This is very important. This involves 1 step, this involves 2 steps. Okay, and these steps are reversible, while this is irreversible. Okay, somebody can say this also involves 1 enzyme, this involves 2 enzymes. Okay, let's look at what the mass scheme says. Good. Similarities: use pyruvate, yes, occur in cytoplasm, make original NAD redox reaction. Differences: lactic acid made in mammal, ethanol in yeast. This is ethanol in yeast. This is yeast. This is for yeast. Carbon dioxide made in yeast. Okay, yep. And then reversible in mammal and irreversible. Okay, let's go on to the next question, guys. The tiger barb pontigrus tetrazona is a South American fish that is popular worldwide as an aquarium fish. Fig 6.1 shows the appearance or phenotype of a normal wild type. This is a normal wild type tiger barb. Okay, aquarium. Do you like aquarium? No, this fish is cute, man. It's beautiful. Look at it. Beautiful fish. All right, that's a white type. You can see, okay, the appearance, phenotype, normal, white type, tiger barb. Okay, let's look at further information. Tiger barbs that shows a white type phenotypes are gold. So white types, please don't forget that. They're giving information, they will ask you for that later indirectly. They are gold with black stripes. Now, tiger barbs that show albino phenotype are gold with white stripes. They don't have black stripes. Okay, gold but white stripes. In 2012, a fish breeder discovered a tiger barb with a new transparent phenotype. Now this has transparent phenotype, not gold. This fish had a transparent body and black stripe. So it must have come from here. Okay, from white type. So it had transparent body and then black stripe. Okay, let's see, guys. The fish breeder crossed tiger barb showing the new transparent phenotype with a tiger barb showing the albino phenotype. Okay, cross two of them. All F1 offspring were white type. Wow, white type is dominant. These F1 offspring were crossed with each other. All right, so they were, all right, table 6.1 shows the phenotype of them in F2 generation. So when the F1 was crossed, they form F2. All right, and the number of fish showing each type. So this is the table, guys. For F2, remember that F1, all F1 showed white type. Okay, so that is it, guys. Can see white type for F2, albino, white. Okay, transparent. So let's go. The fish breeder calculated the new transparent phenotype that had occurred. Okay, concluded, sorry, that the new transparent phenotype had occurred because of mutation. Explain how the result in table 6.1 support this conclusion. It's about 2 marks question. So the guy said that this transparent phenotype was due to mutation. Why is it due to mutation? Okay, pause the video and try. Just rack your brain. Think. Why is it due to mutation? Okay, why is it due to mutation? Write the answer. Okay, and post it on YouTube there. I'll send you chocolate. All right. Now, of course, why it is because number one, you see that the transparent, right, phenotype, okay, is new allele. Mutation introduces new allele, and it's not just that it is new allele, it is also recessive, because when it was crossed, all the F1 offspring were white type. Okay, so that transparent guy is recessive and is new. And if you check also, you find out that, all right, the white type, all F1 offspring, they were white type. And to show that this is mutation, it is transferred to second. This is F2 phenotype, so it is inherited. Okay, so it's genetic, it's inherited, because mutation create new alleles which can be passed on. So explain how the result. I've explained it. Number one, the transparent is a new allele, okay, and is recessive. Number two, it is inherited, passed on. Number three, okay, of course we've said that the white type is actually dominant. Okay, so all F1 were all white type. Okay, that shows that it is of course due to mutation. Yeah, look at it. Transparent phenotype appears in fig. It doesn't appear in, it only appears in F2. That's here. It doesn't appear in F1. So it is inherited, passed down. Yes, it's not environmental. Yeah, so new recessive allele, there's too much. Okay, so mutation is not due to environment. Okay, yeah, so new recessive allele. Okay, let's go on. State the approximate whole number ratio shown by result in table 6.1. Now, guys, don't forget that anytime you do, dihybrid heterozygous crossing, dihybrid heterozygous crossing, for chromosomes, for genes that are not linked, is always 9, all right, is to 3. That's the phenotypic ratio. Okay, 9 is to 3 is to 1. All right, so have it in the mind. But let's go. The state approximate. There's a state the approximate whole number ratio shown by the result. Yeah, that's it. Okay, 9 is to 3 is to 1. That's it. Why? Because heterozygous dihybrid heterozygous crossing for chromosomes that are not linked and there's no epistasis will give you this, right? If there's epistasis, it will change. If they are linked, it will change too. Okay, if there's epistasis to give you 934, if it's linked, then 1 is to 1 is to, okay, 1 is to 1. All right, look at this. Explain what the result in table 6.1 show about genes and alleles that determine the Y type, all right, albino, and the 2 different transparent phenomena. Okay, so you see the Y types are dominant, all right, alleles, albino and the transparent, they are all recessive. That's what it shows. It shows that there are 2 genes that are involved here, and those genes are not autosomally linked, they are not sex linked too. All right, yeah, so look at it. So you see, so 2 genes are involved, all right, so one for gold, one for, okay, black. This mutates to form transparent. Why this mutates? All right, this one has 2 alleles, either gold or transparent. While black stripe has 2 alleles, either black stripe or albino. Do you get? Okay, so 2 genes on separate chromosomes, they are not linked. That's why we're having this result. Not sex linked, autosomal. Each gene has both gene, right? Yeah, they are, both of them have 2 alleles. Yeah, either gold or transparent, either black stripe or albino, or that's white stripe. Okay, albino was recessive, transparent not go recessive, white type is dominant. So that's what it shows. Okay, what the result shows us about these genes. Right, guys. F1 tiger barbs all look the same, but F2 offspring showed variation. The F2 offspring showed 4 different phenotypes. Yeah, describe the process that occurred during meiosis in the F1, all right, fish that allowed this variation to occur. Okay, in meiosis create the genetic variation, guys. Do you remember? What stages of meiosis does this occur? Pause the video and tell us. Answer this question. Okay, pause this video and try to answer this question. It says describe the processes that occur during meiosis. Or for the first one, that create these different phenotypes. Okay, there's reshufflement of allele. The alleles are reshuffled. Is that okay? You can look at what occurs in metaphase 1, which is independent assortment. So the chromosomes, parental chromosomes, align independently. They align independently to each other along the metaphase plate. So because they align independently, so they are, of course, inherited differently. That means that we have different combinations of the alleles in the gametes. All right, so different combinations, and each combination is actually determined by using 2 raised to the power n, where n is the number of genes. Okay, here now we have how many possible, right, phenotypes, which is 2 raised to the power 2, the number of chromosomes. Okay, so you have 2 of them, pairs, 2 chromosome pairs. That's about 4 different combinations, right, for independent assortment. All right. Okay, now of course crossing over can occur, but crossing over only affects genes that are linked. So we're not mentioning crossing over because these genes are not linked. They are on different chromosomes. Okay, it's only independent assortment that can combine them differently. Look at it. Okay, so if you check this, see homologous chromosomes pair up, they form bivalent at prophase, independent assortment, random orientation. Right, it happens at metaphase 1. 1 chromosome, each pair has dominant allele and 1 has recessive. 4 combination permutations. I've mentioned it, worked out example. So they line up at the metaphase plate. Okay, and that is by independent assortment. And then we have different combinations of these alleles in the gametes. Okay, so guys, we're going to stop here. Remember to share this video. This is schedule. So wait for us as we go to the next episode. Thank you. Bye.